Hi, folks. How you doing today?
Doing well. How about yourself?
Good.
Not too bad, thank you.
I'm well, thanks. How are you?
Okay. All right, gang. Well, welcome back. Thanks for showing up before Thanksgiving. I know everybody has other things on their mind right now, and I appreciate that. So we're going to get started with chapter 14 today, but just two quick business items before I do that. Let's see here. So let me remind you, I believe I've said that we do have the final exam schedule at this point, so I'll continue to remind you. Your final exam for this course will be Thursday, December 16th at 12:30. Thursday, December 16th at 12:30 is your final exam. Okay. So I also wanted to just show you something recently that came out of the news. It's a science result that came out of a lab in Massachusetts. And it really doesn't have anything to do with microbiology, but I thought it was significant enough that you would like to see this, because I know most of you are heading down a medical career, okay? And certainly, if you're going to spend any time in the hospital or clinical setting, you're definitely going to come across lots of people that are getting treated for cancer, right? So cancer is a big problem for society. And I just wanted to show a rather interesting and exciting finding that came out of a lab from the Brigham and Women's Hospital up in Massachusetts. So I'm going to share my screen with you and just bring up the news article here. Okay, so go ahead if you would and read that headline. Okay, it says, cancer cells use tiny tentacles to suck mitochondria out of immune cells. So I know that many of you have had anatomy and physiology, so this article can mean something to you, but just to give a little bit of an overview, what you see here in the left picture is a cancer cell. This happens to be a breast cancer cell. Thank you, Samantha, I understand. So this is a breast cancer cell, and what scientists have discovered is that cancer cells can apparently generate these cytoplasmic extensions, which are, in this context, it's being called a nanotube, that has the ability to puncture through the plasma membrane of a T cell. For those that aren't familiar, T cells are short for T lymphocytes, which are part of your immune system and help protect you against infection with viruses and bacteria, but also protect against abnormal human cells like cancer. So the cancer cell is sticking this nanotube through the T cell's plasma membrane, and there's pretty compelling evidence, because they have video on this, where the cancer cell is literally pulling the mitochondria out of the T cell over to itself. So obviously, if the mitochondria is the power pack, the battery pack of the cell, that cancer cell is essentially de-energizing the T cell, leaving the T cell without a capability to produce any ATP. And the mitochondria that's going over to the cancer cell can be used to generate ATP for the cancer cell. So this is a finding that I don't think anybody in the research community expected. This is kind of a shocking thing. It's kind of neat when there's rare moments in life when you find something like this that's like, it makes you sit back and think, huh, okay, so, perhaps being aware of this kind of thing might lead to different cancer treatments. If we can find some medications that would inhibit this nanotube formation, we might have a way of slowing some cancer progression down, okay? So I thought you might be interested in that, even though it's not technically a microbiology topic. Okay, any thoughts or questions? Okay, so folks, before we get started in chapter 14, I just wanted to give you an opportunity to ask about lecture exam 3. Okay, so the study guide was sent out last Thursday. And so I'm hoping you had a chance to at least read over it. Are there any topics on the study guide, or is there any content for lecture exam 3 that you would like to ask a question about?
I have a question. It's Elena.
Okay, Elena.
So, the rRNA, the ribosomal RNA, operon, that one that you told us to kind of review, I'm kind of like, I'm not clear on it.
Okay.
Like
so-
I was reviewing operon and all that area in that chapter, but I'm not quite sure what you want us to be clear on.
Okay, so let me share my screen here. And so here's the study guide that was sent out to everybody, right? So I think you are asking about number 2 here, the graph interpretation question, where the topic is going to be the influence of ribosomal RNA operon copy number on bacterial growth. Okay, so
It's that one. Understand what RR and what a ribosomal RNA operon is and why, how it is important for the growth. I'm not quite clear on that.
Okay. Fair enough question. Some of this, I'm going to kind of hint at things and ask you to do a little bit of research on your own, and you might have to do some research beyond your textbook on this. So a little bit of Googling could go a long way. But remember that ribosomal RNA is a structural component of the ribosome. That's the first thing that we remember that.
Yep, got that.
Okay. We remember from chapter 8 that an operon is a group of genes that are all transcribed from a common promoter. Another way of saying that is that an operon is a group of genes that are kind of expressed together. So the genes of the operon are all made into a piece of RNA at the same time. Okay. And so what I'm asking you to do is do a little bit of reading about, well, let's see, a little bit of reading about ribosomal RNA operons and copy numbers. So what I'll do is do a live Google search here on this topic with you. And let's see if we encounter any things that might be helpful. Okay. Ribosomal RNA operons and bacterial growth. There we have some links to multiple ribosomal RNA operons in bacteria. Here we have ribosomal RNA operon copy number reflects something with ecology. Okay, ribosomal RNA operon copy number reflects ecological strategies of bacteria. So, Elena, do you think you could do that kind of Google search at home?
Yeah, that's why I started. I was kind of looking, outside of the text and the notes, trying to get a kind of an understanding of it a little bit more.
Beautiful, and that's exactly what I want you to do. So you're on the right track, okay? And so please understand that if I go back to the study guide, I am going to ask you a couple basic questions about the biology of ribosomal RNA operons and why it is important for the growth of the cell. But after I ask you that basic biology question, then the rest of the essay is going to be about interpreting a graph. So you don't have to be an expert on ribosomal RNA operons. What I want you to do is interpret the data in the graph. It's just that the data in the graph happens to be on the topic of ribosomal RNA operon copy number.
Okay, got it.
So
Thank you.
it's okay. I have a little suspicion, and you can confirm or deny, that you might be sitting at home thinking, okay, that didn't help me as much as I hoped, okay? And so I am intent.
I just didn't, I really didn't want to know as to in what depth you wanted us to, to know this. I know it's going to be in a graph form, so it's more the graph. So now what I'm interpreting is that when I look at the graph, it's kind of more or less to understand what is going on in the graph. You know what I mean? Like when I look at the graph, that I could be able to understand it in terms of the ribosomal RNA and what it does and how it affects the cell and all that.
Exactly, right. And so we've done some homework problems from the workbook where you did some graph interpretation. So this is going to be along the same kind of lines of the workbook practice questions you've had this semester. So, I think if you understand a definition of what a ribosomal RNA operon is, and you could write out maybe 1 to 3 sentences on why it is important for the growth of the cell, then you will be able to handle the biology part of that essay question. And then the rest of the essay question is really about, well, can you interpret this graph and draw some meaning from the data that's being presented to you? So, okay. Are there other aspects of this question, Elena, or anybody that you would want me to go a little bit further in my hints on?
So what we're saying is you're going to show us a graph and we have to write an essay in reference to what we see in the graph.
Well, remember, it's not going to be a traditional English essay like you're in an English literature course. It's going to be, here's a graph that illustrates some data, and I'm going to ask you a series of questions about the graph, just like I did with the workbook homework. I'm going to ask you some questions. What does this mean in the graph that you're looking at? Okay.
Okay, that's clear. I'm just trying to prepare myself because I know your essays are a bit different. I've experienced different essays with you.
Okay. And so,
Yeah.
your best preparation for these kinds of activities is to go back and review the workbook problems that we did together. Make sure that when you go back through them, you understand the answers that we provided or that you created for yourself. So make sure that that's flowing nicely for yourself. And then if that is flowing nicely for yourself, you should have confidence that, yes, I can interpret data in a graph. Now, I just want to be sure that I understand the basic idea of what a ribosomal RNA operon is and why it is useful to the growth of a bacterial cell. Okay. Okay, anything else, folks, about any aspect of the study guide or content that you might be stressed with on the Tuesday, a week from today? Okay.
I am. I was trying to do the extra word problems that you sent out with the last lecture notes. I'm just having a hard time trying to put it like put the log number. Like I see your answers and I see how you got the answers for the logs, but I just don't know how to put it in my calculator for me to get the same answer that you got.
Okay, sure thing, Yasmin. Tell you what, that's probably an individual concern with your calculator, something we can deal with in like 2 minutes. So just hang on with me after class today and we'll wrestle through that, okay?
Okay, thank you.
Any other questions or concerns?
You wanted us to understand the glycolysis, the whole Krebs cycle and all that. So this study guide is just more or less a brief description of kind of what's going to be on the test. But outside of this stuff, there's other things that can possibly pop up.
Okay, so just to help you, because I know there's a tremendous amount of content, particularly in the metabolism chapter. So first off, remember, I will not ask you any questions about photosynthesis from chapter 5. That will not be on the test. Also realize that, yes, in a perfect world, I would like my students to understand glycolysis, Krebs cycle, electron transport chain, all that kind of good stuff. But we don't live in a perfect world. and I'm a little bit of a realist. So what I'm telling you is the only essay question you will get from chapter 5 truly is the question about the electron transport chain. That's where you're going to be challenged the most on your understanding of chapter 5 is, do you understand the electron transport chain? Now, that being stated, of course, I could ask you some questions in the multiple choice and fill in the blank that have something to do with glycolysis, or has something to do with the Krebs cycle, or has something to do with oxidation and reduction, right? I mean, I could ask those sorts of questions in the typical multiple choice and fill in the blank questions.
Okay, that's fair.
Okay, anything else? Okay, so, you will have an hour and 25 minutes to take the exam, but also take note that in terms of the math problem, I'm giving you 15 minutes to get to me, right? So I'm kind of sneakily giving you another 10 minutes to actually work on your math problem and get me an answer if you're struggling with it, okay? So I'm trying to be reasonable about the time I'm giving you to take your exam. Okay. Okay, so any other concerns? When you take this exam on Tuesday next week, you want to be sure you're using good test taking strategies. And what I mean by that is when the questions come up on class marker, answer the ones that you feel confident about right away. And if you have a question in front of you you don't feel confident about, write that down on a piece of scrap paper and move forward in the exam and answer all the other questions that you feel confident about. Once you've run through the entire exam once and answered things you're confident about, then look at the questions on your scrap paper that you intentionally skip because you're not confident on them. Then start going back to them and working your way through it. And of the questions you're not confident about, well, you're going to have to prioritize. Which ones do you think you can get some partial credit on and spend your exam time on that? Okay, so you want to take the test from an intelligent perspective of how do you take a test properly to maximize the amount of points you can earn in a given time that you have available. What I'm saying to you is this. If you get to the math question that you know is going to be on here and you look at it and it really scares you, fine. Then maybe you spend most of your time on other questions on the exam to get as many points on other parts of the exam as you can. You need to be honest with yourself. I can do this now, or I can't do it now. And spend your time where you will get most credit, okay? I know you're up for the challenge. The part of it is you get yourself a little bit psyched out on an exam with the time crunch, okay? So anything else before we continue?
Yes, I actually do have a question. I was looking at the number 6, the topic of the disk diffusion plates and zones of inhibition. I didn't see it on the lecture notes. I just wanted to make sure that it was something that we needed to get out of the book.
Yep, this was not in the lecture, Yasmin. Truth be told, if we had a normal semester, you would have gotten part of this topic in lab. But since we're not doing a normal semester, this was not presented in lab. However, I believe if you read the content of the chapter around figure 20.17, and perhaps even do a little bit of Googling about disk diffusion plates and zones of inhibition, you really should be able to understand what the basic idea of this is and be ready to do okay on the exam.
Okay, so it would just be like a general background of what it is.
If you understand what it is and what it means, then you'll be able to answer my essay question.
Okay. Thank you.
Anything else? Okay, folks, we'll get started, but I certainly don't expect you to work on Thanksgiving Day. Please take some personal time for yourself. I want you to take personal time for yourself. But obviously sometime over the holiday weekend, you are going to really want to do some studying for this exam. And remember, when we come back on Monday, I do have office hours Monday morning. I'm happy to spend time with anybody that has a problem. If you shoot me an email, over the weekend, I probably won't answer it until Monday morning, but I will answer it on Monday and get back to you before the exam, okay? So there's still some time that you can reach out to me if you really need help. Okay. All right, folks, let's get started here. Chapter 14 is basically about epidemiology. So in my textbook, chapter 14 starts page 393, 394 in that vicinity. Okay. And before I get started, I typically get started with some basic definitions from this chapter, but I'm going to start with a YouTube video about a topic here. And, when we talk about disease in our human bodies or disease in an animal, right? Some people do care about when animals get sick. When we talk about disease, it's kind of a double-edged sword, because we know when we're not feeling well, and we tend to take for granted and not put much conscious thought to when we are feeling well. So what I'm saying is, if we're going to understand what the absence of health is, we probably want to understand what the presence of health is. And so when it comes to infectious disease, those disease processes that are at least somewhat correlated to the presence of a microorganism, a fungus, bacterium, protozoa, virus, what I'm saying is it would be very helpful to the medical community if we knew what types of microorganisms are naturally found in and on the surface of the human body that are present even in people that we feel are healthy, that are feeling just quite okay. All right. So there is a project that your book references in the early reading of this chapter called the Human Microbiome Project. And I'm going to let this video kind of explain the purpose of this project and give you a little bit of visualization on it. I'm going to put the video link in your notes here. Okay, so you can watch it again if you want to. Well, let's let the video roll through for about 2.5 minutes or so you can get the gist of what's special and significant about this project, and then we can relate it to the content of chapter 14, okay? All right, so let's get this rolling here. And here we go.
My name is Curtis Huttenauer. I am an assistant professor in the Department of Biostatistics at the Harvard School of Public Health, and I work on the human microbiome, which is the collection of microbes, single-celled organisms like bacteria, viruses, fungi, and many other organisms that we carry in and on our bodies. There are about 10 times as many microbial cells in our body as human cells. Our skin, our mouth, our airways, every exposed surface in the body carries its own naturally occurring microbial community, which typically has evolved with us to promote health and protect the human body in that particular environment. Our group helped with one of the main consortium analysis efforts in order to interpret these sequences as a map of what our normal microbial variation is. So this really gives us a definition of the boundaries for over a dozen different microbial habitats associated with the human body, of what kinds of microbial communities can be normal in health. The microbes associated with our bodies give us another way to potentially assess lifelong risk of disease or immediate risk of disease outcome. What we're learning right now is how better to use our microbes to predict the risk of autoimmune or inflammatory disease onset, IBD, diabetes, diseases in that family, and how to predict the risk of their outcome. So for example, the onset of a Crohn's disease flare using not our DNA, not our own genes, but our microbes and their genes and biological activity. One of the reasons, or one of the areas why a lot of people in this field are very interested and very motivated about the topic right now, is that the microbiome is part of our environment that might help to explain the last 50 years or so very rapid increase in chronic non-genetic disease. If environmental exposures are part of what's causing this increase in obesity, in diabetes, and other chronic conditions, and that's something that is affecting our microbes in a way that can be treated or prevented or used to improve health, that's a huge motivation and something that needs to be worked on now as quickly as possible in order to help ameliorate the rise in chronic disease and obesity. It's a very exciting time now to be working on both computational biology and DNA sequence analysis generally, and the microbiome in particular. These are both very new areas that are changing rapidly. There's a lot of work to be done. And I'm looking forward to seeing how the Harvard School of Public Health, our students, our group, and others carry out research in the area.
Okay, folks, so that's just a quick little introduction, and let's just put a summary statement into your notes about what the purpose of the Human Microbiome Project is. So I would, we could define it a number of ways, but, so let's just get something that seems to be a useful starting point for this conversation. The purpose of the Human Microbiome Project, and believe me, it's not just a project carried out by Harvard, although Harvard's got its hands in just about everything because it's got lots of money, right? The purpose of the Human Microbiome Project is to survey and catalog the different species of microorganisms that are found on and inside the human body in different locations, right? In the bloodstream, in the genitourinary tract, in the respiratory airways, in the ear, any surface that you can conceive of inside the intestinal tract, any area you can conceive of. Let's survey and catalog the variety of species of microorganisms that are found in healthy people, okay? The purpose of the Human Microbiome Project is to survey and catalog the variety of species of microbes that exist in and on a healthy person's body. Okay, and this is a nice segue into some basic definitions and terminologies of this chapter. And so I'm going to start with the phrase, or with the term disease. Okay, so disease is typically defined as any state or condition other than a healthy one. Okay, and there's some points about this. There are some disease states that are extremely obvious to recognize, right? I mean, if you're vomiting into the toilet, well, you must be sick. That's not a natural thing. You're not supposed to normally do that, right? So, obvious signs of fever, obvious signs of vomiting, shivering, things like this that are abnormal indicate some kind of problem. So the person is not healthy in that moment. If you got a broken bone and it's protruding from the skin of your leg, ouch, that's not healthy at all, right? All that kind of stuff. There are some things that is very obvious when we have the absence of health, and we call that absence of health disease. However, there are more subtle things, right? Think about all the very subtle things of peculiar psychological behavior. Think about the people in your life that you might know I mean, I'm sure we all know one or two people that were like, well, they might be a few cards short of a full deck, right? I mean, there's just something strange in how they function, at least from our own viewing perspective and our own prejudices and biases. And so what I'm saying is that identifying physical disease is pretty obvious, at least more obvious than identifying necessarily mental health issues, okay? Okay. All right, let's continue. The term pathogen. Pathogen refers to any disease-causing microorganism the term pathology. Pathology is the scientific study of disease. Now, when we talk about pathology, there's typically three subtopics that come into focus, right? If you are a pathologist, somebody who studies disease, there's typically three areas of concern that you would be interested in with your work. First would be etiology. Etiology refers to the cause of the disease. And you think, you know, in today's modern world, we think that that's a fairly easy thing to determine, but it's not always easy. And let me refer you back to the story of the 1918 Spanish flu pandemic that we started the course with some months ago. Remember that in 1918, the best scientific minds of the day felt that the influenza outbreak was caused by a bacterial microbe called Haemophilus influenza. And we now know that that's laughably false, right? We know that that's definitely a viral disease. But back in 1918, they did not have the technology to do anything to actually measure the presence of a virus. So determining the cause of a disease is super mega important if you don't want to waste time, resources in terms of person power, the efforts of men and women trying to combat the disease, as well as financial aspects of providing necessary resources, right? I mean, if we have influenza and you're cranking penicillin into somebody's body, that's not going to be helpful, right? Because penicillin treats bacterial disease. Influenza is a viral illness. So identifying the etiology is extremely important. There's also pathogenesis, okay? Pathogenesis is a fancy word which represents the stages in which the disease develops. Pathogenesis refers to the stages of disease development. You know, thinking about a hypothetical infection of any kind, maybe you get infected with a hypothetical microbe of some kind, and maybe the first stage of the disease is you develop a fever. Maybe the second stage of the disease is you develop a rash. Maybe the third stage of the disease is you vomit. Or maybe you could shuffle those things around into different stages. Maybe the first stage of the disease is you vomit, and the second stage is you develop a rash, okay? So the stages of disease progression are something that a pathologist is concerned with. And finally, a pathologist would be concerned with identifying the structural, and I mean, well, by structural, I mean the anatomy of your body and how it changes. Identifying the structural and functional changes that take place in the human body as a result of the disease progressing. Pathologists are interested in understanding and describing the structural and functional changes to the human body that occur when disease is present. Okay, so these are some talking points, and hopefully, thinking about what the human microbiome video was telling you about, you can start to see what the focus of this chapter is really going to be. So in terms of, what kinds of microorganisms are normal in the body? Well, that's still a largely unfinished question. We don't have a final answer on that, and we may never have a final answer on that. But from work with the human microbiome and even work before that official project got started, microbiology has become aware of various species of microbes that are normal inhabitants of healthy persons. And we're given a subset of those microbes in table 14.1. So I'd ask you to turn to table 14.1. I'm not going to catalog this in your notes because your book suffices for this. But let's highlight a few things. When we say normal microbiota, we are referring to the microbes that live in and on your body when you feel fine, when you don't feel sick. This refers to the community of microbes that live in and on you when you feel healthy. Okay, sure thing, Taylor, thank you. So remember, folks, we've said it again. The video actually said something in reference to this. We know that the human body is made of roughly 50 to 100 trillion human cells. And the best estimates that science can provide is that for every one human cell making up your anatomy, you're going to have at least 10 microorganismal cells of various types, fungi, protozoa, bacteria, viral particles, things like that, that are also part of your body as well. So let's talk about some examples here from table 14.1, and I'll start with the skin. Okay, it's unfortunate we couldn't get together for lab this semester because we would have cultured microbes from your skin, and it's kind of a fun thing to do. So from the skin, I'm going to highlight that it's very common to find members of the bacterial genus Staphylococcus present. Okay, and so please understand that Staphylococcus is going to include that famous guy, Staph aureus, right? So this includes Staph aureus. But please understand that Staph aureus comes in many subtypes and varieties, just like we are all humans, right? We're all humans, but none of us really look the same, right? And we can break it down in terms of, with humanity, we could break it down in terms of nationality, right? North Americans, Europeans, South Americans, things like that. So there are different subtypes of Staphylococcus species. And what I'm saying is, it is very common, even in healthy human beings, to have members of the Staphylococcus bacterium on the surface of our skin. They are a normal, natural inhabitant. Most times they don't cause us harm. The exception to that rule, of course, is the MRSA variety, which does cause us harm. Okay. Most subtypes of Staph aureus do not harm us, but the notable exception is MRSA, right? So MRSA is short for methicillin-resistant Staphylococcus aureus. Which we all know horror stories from various, medical situations with that. Okay, So Staphylococcus is a common member of the community of bacteria that live on our skin. For our eyes, it might be an uncomfortable idea to think about this, but our eyes represent a surface, and like the video said, any surface of our body is going to be covered in microbes. There is a bacterial genus by the name of Corynebacterium that lives on the surface of our eyeballs. And as far as we can tell, when I say we, I mean the scientific community, as far as we can tell, the Corynebacterium don't really do anything for us, and they don't do anything against us, as far as we can tell. There seems to be no positive or negative effect to Corynebacterium on our eyeballs. Now, of course, there might be a very subtle effect over many years of time that we just are completely oblivious to. You know, that is possible, right? Because it's hard to do scientific studies studying these things that last for decades. I mean, what if the presence of a Corynebacterium on the surface of our eye over 30 years, 3 decades, promoted cataract formation? What if that was true? Well, we have no idea of knowing if that's true because nobody's done a 30-year study on this. Okay. Let's see, nose and throat. Nose and throat, it's very common to find members of the bacterial genuses Haemophila, Haemophilus, and Neisseria. Okay. Now, both Haemophilus and Neisseria genuses include species of bacteria that do sometimes cause disease. However, 9 times out of 10, our body's own secretions, like in our nasal secretions and in our throat secretions, the secretions contain molecules with antibacterial properties that keep these microorganisms suppressed enough that they don't usually cause us harm. Okay. Let's see, the mouth, okay. Well, you know, a lifetime ago, when I was a little boy, I took some martial arts and very much enjoyed it. And I remember my martial arts instructor saying, if you're going to punch somebody, don't punch him in the mouth, because the mouth is just a zoo of microorganisms that if you cut your hand on somebody's tooth while punching them, you could really get a horrible infection. I just remember that as a little boy. And there's a lot of truth to that. The mouth is just it's just like your mouth is like the microbial equivalent of going to the Philadelphia Zoo. There's just a whole treasure trove of microbes that live in our mouth, okay? So it's very common to find members of the Lactobacillus genus, which is a bacterial genus. And it's also common to find members of the Candida genus, which is actually a fungus. Okay. And again, 9 times out of 10, these things don't really do too much to us, as far as we can tell, in terms of causing disease. In the large intestine, of course, I would be remiss if we didn't mention E. coli. And you may know from things like A&P that E. coli is capable of manufacturing certain vitamins that our own cells cannot manufacture. Some of the B vitamins and vitamin K are manufactured from the E. coli in our large intestine, and those vitamins get into our bloodstream and help our bodies function better. Okay. And so finally, we'll mention the urinary and reproductive systems. Very common to find members of the Pseudomonas genus in both of these systems. And at least in the female reproductive tract, it is common to find a protozoal microorganism by the name of Trichomonas vaginalis. Okay, when you look at the phrase Trichomonas vaginalis, obviously the species, the specific epithet term, includes the word vagina. So this microbe was named because it was first cultured and identified out of the female reproductive tract. Okay, so folks, you can see we could quickly get a ridiculous almanac-sized version of this table, and I didn't want to do that, but I wanted to be sure that you are at least looking at table 14.1 to see that there really is a treasure trove of microorganisms that live in and on our bodies, and we're still trying to figure out a lot about this. Okay, any questions so far? Okay, next topic. We're going to jump to the next page. If you're looking at table 14.1, then the next page has a major section which talks about the relationships between the normal microbiota and the host. Okay. And so I suppose I can bring this down onto the next page and start it fresh. Okay, so there's going to be some terms here that we should describe. First is microbial antagonism. Okay, now I don't know about you, but if you grew up with a brother or sister, usually that relationship is fairly antagonistic. I mean, I wouldn't say it's always antagonistic, but if you grew up with a brother or sister, you might ask yourself, did your brother or sister ever get on your nerves? Or did you ever get on your brother and sister's nerves? I suspect the answer is yes. But even though you might get on your brother and sister's nerves, I also think it's a fairly common, at least I hope a common experience, that while you might irritate your brother and sister, I bet you're probably one of the first people to stand up and defend your brother and sister if some person outside your family is in any way threatening your brother or sister. Okay, so microbial antagonism is a two-sided coin here, is what I'm trying to say. Microbial antagonism refers to a situation in which the presence or growth of one type of microbe interferes or blocks the growth of a second type of microbe. Okay, and so one of the pieces of information that scientists are searching for with the Human Microbiome Project is to figure out which microbes really belong on the surfaces of our bodies in a healthy body, because if we have somebody that's sick, maybe they're sick because a different kind of microbe has captured that surface, so to speak, and we need to do something to replace that community of disease-causing microbes with healthy microbes. We need to identify what those are. Okay, in terms of microbial antagonism, this is one of the reasons that if you're prescribed an antibiotic, excuse me while I read the chat window here, Kit is asking, is that basically what probiotic yogurts do? Yes is the quick answer to that question, Kit. Yes, probiotic yogurts, and we're going to talk about probiotics in this chapter. You know, they tend to repopulate cavities, spaces, surfaces in the body with healthy microorganisms. Yes. Okay. So one reason you should complete a full therapy of antibiotic treatment prescribed by your doctor is because if you don't do that, you know, antibiotics are not perfect. They're not perfect at just killing the bad microbes. They're also going to kill some of the good ones as well. So when you're given an antibiotic prescription, you're given a prescription with the best available scientific guidance, and that's not perfect because science is not perfect, but you're being given a prescription that, as far as we can tell from a science perspective, should knock out 99.9% of the bad guys. But if you stop that prescription early because, you know, you've been given 14 days of antibiotic and you feel good on the 10th day, so you stop taking it, there's a real chance that you haven't killed off all the bad guys yet, and they, through microbial antagonism, might be able to beat up the remaining good guys and take over that area of your body and cause an even worse problem later on. Okay. Yeah, let me get this in your notes here. One reason you should complete a full therapy of antibiotic treatment prescribed by your doctor is because if you stop treatment prematurely, microbial antagonism of unhealthy microbes may be able to supplant, you know, to take over the healthy microbes and create an even worse problem, worse infection than you started with. Okay. All right. So, you know, think about Clostridium difficile infections in patients, right? I mean, a lot of people develop a C. diff infection, and sometimes it's because they didn't finish their antibiotic therapy properly, and then the C. diff bacteria were able to overwhelm the E. coli in the intestine, and now you got an even worse situation to deal with. Okay, so let's talk about various types of relationship terms that are encountered in microbiology. So the term symbiosis. The term symbiosis refers to living together, okay? And, you know, from a human perspective, we might think about a symbiotic relationship with our boyfriend or girlfriend or spouse or something like that, but please understand that the term symbiosis, at least in science and at least in microbiology, does not imply a quality of good or bad to that relationship. You have microbes living together, that's a symbiotic relationship. We're not talking about whether the relationship is a healthy one or a good one. We're not talking about the quality of the relationship. This term does not imply a quality to the relationship of good or bad. Okay. Now, there are some terms that do imply a quality to the relationship. There is the term commensalism. Okay, commensalism. So right now, I'm still on page 397. You look at figure 14.2 here. You see some of these words that I'm putting in your notes from figure 14.2. Commensalism. This is a type of symbiotic relationship in which one microorganism in which one organism benefits and the other organism is not benefited or harmed in any way. So the example that we just recently talked about is the corynebacterium on the surface of your eye. Okay. This is a type of symbiotic relationship in which one organism benefits and the other is not impacted positively or negatively. Okay. So as the example here, corynebacterium on the surface of our eye, clearly corynebacterium benefits because they now have a home to be on. That's the surface of our eye. And, without a doubt, some of the fluids, some of the tears that go across the surface of our eye to maintain the lubrication of that space contains micronutrients that bacterium can actually live off. So the corynebacterium are benefiting from that environment. But as I said, as far as we can tell, the human body is not impacted positively or negatively in that regard. So let me just copy that down into your notes here. All right, and let me also reference figure 14.2 here so that you know to look at that during your study time. We have mutualism. Mutualism is a symbiotic relationship in which both organisms benefit. And an example here would definitely be the E. coli in your large intestine, right? E. coli gets to live and thrive off the undigested nutrients that your own body doesn't want, and therefore is passed from the small intestine to the large intestine. And we benefit because the E. coli then makes some of the vitamins that helps our cells work better. Okay. And finally, there's also parasitism. That one probably doesn't sound very nice, and it's not very nice. This is the type of symbiotic relationship in which one microbe benefits at the detriment or expense of the other. So in this scenario, think about your favorite pathogen. Okay, if you have a favorite pathogen, that's an example of a parasitic relationship in the human body. So, pick your favorite pathogen. And so as an example, we could say, the bacterium that causes tuberculosis, Mycobacterium tuberculosis, that's the name of the bacterium, right? It lives and thrives in your lungs, and we're not too good because of it if you were unfortunate enough to contract that. Okay. All right, folks. So, I feel that this chapter is actually a very accessible chapter. And what I mean by that is there's not a lot of hard chemistry or math ideas to understand in this chapter. There's just some ideas, some words, some vocab for you to learn. And so I think this is an accessible chapter. And I don't think you should worry about it too much. I think as you read it, you'll pick up the things that are most significant. Let's talk a little bit about probiotics. Okay. Okay. Probiotics are living cultures of microorganisms that are designed to provide benefit to the human body. And that refers to your statement in the chat window, Kit, about the various kinds of yogurts that exist out there. Okay. Probiotics are living cultures of microbes designed to provide benefit to the human body. You might have heard that term prebiotics, right? Prebiotics are not cultures of microbes, they are cultures of nutrients. They are media and food recipes of nutrients designed to encourage the growth of healthy microorganisms in the body, okay? So you might have heard of that as well. Probiotics are these living cultures of microbes designed to benefit the body. And so let me give you a couple examples here. Okay. Let me see here. I have a couple of notes that I want to share with you. I'll type a question into the chat window. Have you ever seen or used an over-the-counter probiotic called Align? Okay, well, let's see if I can get an image up here real quick in the chat window in Google Images. There we go. Maybe you've seen some of these boxes at the supermarket, right? The Align probiotic. Align, I believe, was one of the first ones to come on the market some years ago. I couldn't even tell you how many years ago it's been now. Okay, so thanks for the comments in the chat window. I appreciate that. So Align probiotic contains a strain of bacteria that's been patented by the company that manufactures Align. So please understand, companies like to discover things that Mother Nature has created and then develop a patent and get, money for it. They love to do that. Whether that's ethical or not is a bigger philosophical question we can argue about at another time. Okay. So the Align probiotic, take a look for it next time you're going through the grocery store. This contains a patented strain of a bacterium called Here it is, Bifidobacterium infantis. Bifidobacterium infantis. And there's a numeric designation after that. 35624. Okay. So, what this is suggesting is that whatever company manufactures Align, they screened at least 35,624 subspecies of Bifidobacterium bacteria, probably from the poop of infants. The poop of infants that they were given access to on a hospital or clinical setting, okay? And they screened for a particular subspecies of this bacterium that had the characteristics that they were looking for, such as trying to calm down inflammation in the digestive tract or improving the regularity of your bowel stool, your bowel movements and things like this, all right? So each pill or each capsule of this Align probiotic contains at least 1 billion of these bacterial cells present that you're passing into your body, okay? A second example would be, and this one kind of speaks to Kit's comment, would be Dannon's Activia yogurt. Okay, Dannon's Activia yogurt contains a species of bacterium called Bifidobacterium lactis, DN173010. So obviously, to the company that's manufacturing these products, they do care about the microbiology of the product that they're manufacturing. They got to keep consistency for good results. But, to the average consumer, typically folks don't care too much, right? You've just been through a bad bout of diarrhea, or you've just come off a strep throat infection, you've been on a Z-Pack for 7 days or whatever, and the doctor says, hey, go pick up a probiotic at the supermarket, and it will help your body return to normal a little bit quicker, okay? All right. Any thoughts or questions about that? Okay, next topic, opportunistic pathogens. Opportunistic pathogens in my textbook are page 398. So an opportunistic pathogen would be any microbe which creates disease when a particular circumstance changes. Okay. This refers to a microbe that does not normally create disease unless a special circumstance is presented. Okay. So let me give you an example here. Okay. Escherichia coli in your large intestine. It's supposed to be in your large intestine. We want it to be in your large intestine because when it's in your large intestine, it does things that benefit your body and helps keep you healthy, such as making some of the vitamins. However, if for reasons that are not always obvious or clear, E. coli is able to relocate from the large intestine to, say, your cerebrospinal fluid, then E. coli is going to cause meningitis and it could possibly kill you, right? So the location of the microbe matters. And as I said, E. coli is not normally a pathogen to us, but if it gains access to certain other areas of your body, which we don't normally want it to be in, then yes, it can make you horribly ill. E. coli is a good companion. E. coli is found in the large intestine and benefits human health when it stays there. Okay. When E. coli gains access to other regions of your body's anatomy, such as the central nervous system, then it can create meningitis and be deadly. Okay, let me give you another example here. Example 2. Okay. If you've been on planet Earth for a while, there's a good chance that whether you knew it or not, you were interacting with someone that was, fighting HIV. HIV's been around a long time. It turns out that there is a fungal pathogen that typically does not make healthy adults sick, but when that pathogen finds itself in an adult body that is immune compromised, such as you would have with an HIV patient, promoting the development of AIDS, acquired immunodeficiency syndrome, then this fungal pathogen actually does cause problems. Okay, so let me document this for you. There is a fungal parasite called pneumocystis giroveci. Let me spell that, Jiroveci, pneumocystis giroveci, that does not normally cause disease in healthy adults. In immune compromised patients, such as HIV infected persons, this fungus does. This fungus causes illness, typically a very, very bad pneumonia. Okay. So there have been cases in the clinical setting, folks, where, a patient went in to a doctor complaining that they felt bad, with pneumonia, and cultures are sent to the lab, and they find this fungal parasite, pneumocystis, and the person, his or herself, has no idea that they're actually infected with HIV. So then the doctor goes and talks to them and says, hey, from a statistical perspective, if you have this fungus in you, we kind of know that it's kind of like a canary in the coal mines, kind of a warning signal that some percentage of people with this fungus also have HIV. Would you allow us to please do a blood test for HIV so we can check this out? Okay, so that unfortunately is some manner in which some members of our population have discovered that they're actually HIV positive. Okay, thoughts or questions right now, folks? Okay, taking a quick look at our time here. We don't have much time left, but at least I can get started with the next major topic. The next major topic is the foundation figure 14.3. This is Koch's postulates. Figure 14.3, Koch's postulates. Okay, so you may recall the 1918 Spanish flu story. And I said just a little bit ago in today's lecture that in 1918, doctors and scientists thought the influenza outbreak was caused by a bacterium. Okay, Robert Koch of Koch's postulates fame was one of the prominent doctors, a German physician, who promoted the idea that influenza was caused by a bacterial pathogen. Well, he was wrong. But even though he was wrong, he still did many other good things in the history of medicine and science that made him famous. And there's the Robert Koch Institute in Germany that is a big famous institute for biomedical research in Germany even today. Okay, so let me just document a few ideas here. We'll stop. You'll be on your way to Thanksgiving. Okay, so Koch's postulate were named after the German physician Robert Koch. Okay. Koch incorrectly identified the cause of Spanish flu, but he redeemed himself because Robert Koch did correctly identify the etiologic agent of anthrax. which is the bacterium Bacillus anthracis. Okay. So we'll end on this note. Koch's postulates, which we're going to have to go over in some detail after Thanksgiving, after we get past the exam. Robert Koch's postulates represent a series of methodical steps, a framework that a scientist must follow in order to firmly establish that a specific microorganism is the cause of a disease. Okay, so these postulates represent a systematic way of providing evidence that a specific microbe is the cause of a specific disease. That sounds like that's an obvious thing that has to happen, but as we'll share with you after Thanksgiving, that's not an easy task to achieve, to actually prove that a specific microbe causes a specific disease. Okay. All right, folks, I'm going to stop talking. I appreciate your time and energy today coming out before Thanksgiving. If you need to hang on for anything, Yasmin, we should talk about your calculator issue. Otherwise, folks, have a wonderful Thanksgiving. Enjoy some time with family and friends. I'll see you next week. Good luck on your test on Tuesday, okay?
Thanks. See ya.
Thank you. Have a good night.
You
Thank
too.
you. Happy Thanksgiving.
Happy Thanksgiving. Bye-bye.